Welcome, everybody. Thank you for being with us today. We are broadcasting live from Park City, Utah, where I am. Rene, where are you, in Texas?
I am in Columbus, Ohio.
Columbus, Ohio. And Peter, you're in South Africa.
I'm in Cape Town, South Africa. Yes, hello.
Yeah. Welcome. Thank you, guys, for coming back for a second time. I've been the beneficiary of watching your progress since the last time we did this. And I'm delighted to say that there's been just amazing things that have happened. And as you'll see going forward in this presentation, what those things are. And it's we just had a very inspiring conversation prior to me hitting live here. So you all will be able to enjoy what I just enjoyed shortly. So we can't hear or see you. We know you're there. We encourage you to ask questions as they come to mind, as we have a lot of people coming on today. If you post them in the Q&A or the chat box, we'll get to as many as possible. Just to be super clear, we'll be putting you in direct touch with Rene post-webinar, so you can have conversations. Also, to be clear, this is our intent here is to raise money for Neurxstem, and so the discussion is going to be around that raise. Also, if there's anybody in your world that would benefit from getting to know what you find out today, and what you could potentially learn more, you're so welcome to do that, and make sure that you know that you can communicate directly with Rene and company post-webinar and share it with those that might benefit as well. If it's okay, we'll be posting the recording post-webinar on the Family Office Insights YouTube channel. And better yet, if we can put it on, Rene, your website, that would be even better. So with that, thank you all again for being with us today. And thank you, Peter and Rene, for coming back once again. So with that, Rene, you want to do a little background?
Yes, thank you, Arthur. It's absolutely a delight to be back. This is for the third time. And hopefully in this third version, I will treat you to something even more extraordinary about the science we do and how it opens the door for space explorations. So I am the CEO of a company that is a predictive and precision functional genomic medicine company, brain medicine company. And I emphasize the word functional because we don't sequence genes. We look at the functions of genes. And its primary goals are diagnosis and treatment of brain diseases and disorders. I'm going to reintroduce our incoming Chief Operations Officer, a phenomenal gentleman. He's a Rhodes Scholar. He's a fellow of the Royal Society of Medicine. He's built businesses. And he's going to give us a top-down summary of the investment opportunity. Peter, the stage is yours.
Thank you very much, Arthur and Rene. Great pleasure to be here. Very briefly, I'm a retired, semi-retired professor of pathology. I have a medical degree, a PhD, and an MBA. And I met Rene when I was acting on behalf of a Middle Eastern family office located in Oxford, England, where I was mentoring Rhodes Scholars, medical Rhodes Scholars, who were spinning off their PhD companies. I went to an invited investor seminar in London, and I attended some meetings, and I was astounded when Rene Anand stood up and said something quite fascinating about functional genomics. In other words, you guys are all chasing genes as gene jockeys. I'm chasing the whole network behind the genes, the way the genes work together, a whole different level of operation. So to cut a long story short, this was 5 years ago. I sort of gave up my day job, and I decided to put my considerable weight behind Rene. And we've been evolving this amazing opportunity, which I'm going to share with you very quickly. Some of this is in the slides. It's very avant-garde, it's very technical, it's very scientific, and very exciting. But basically, we're going to make the case for investing in Neurxstem next phase clinical trials represents an exceptional and compelling investment opportunity. Potentially, this is a unicorn with an addressable market of 150 to 250 billion. And there are 5 pillars to what Rene has put together in the last 10 years. The first is that we have US patented human brain models. Now, it's very important to understand this. Not patient. These are living laboratory cells grown in Petri dishes, which are taken from patients with brain disease. It's human patient-derived tissue growing in vitro. And so it is the real thing. So somebody with Alzheimer's, somebody with progeria, someone with premature aging, for example, or autism, Rene can take a skin cell, he switches off all the genes that are not brain genes, and he switches on all the brain genes, and he grows an organoid, which you'll see a picture of, which looks exactly like a human embryo up to the age of about 3 weeks. It has a forebrain, a midbrain, and a hindbrain. It has light-sensitive areas like the retina. It has electrical impulses like an EEG. And to all intents and purposes, it is the neural tissue derived from that patient. And then even more amazingly, he can use novel drugs to examine this tissue using genomics and metabolomics and proteomics, and find out which genes are lighting up in the abnormal patient versus normal. And then when he adds the drug, which genes are either upregulated or downregulated. And that tells us roughly which neural networks are working. And therefore, by going upstream, we can target the beginning of the cascade of events rather than the end stage, which other people are studying. So in addition to the patented model, we've got novel drugs which show efficacy, and we'll show you some examples for Alzheimer. We've got diagnostic biomarkers which are present as early as birth. For Alzheimer's, this is a completely new concept. It's not happening at the end of life, it's happening at the beginning. And so we may be able to interfere with the process and mitigate, slow down, or prevent. We also have a huge biomarker soft bank, which has been patented for most of the major human brain diseases: Parkinson's, autism, certain forms of cancer, progeria, and of course, Alzheimer's diseases. And this in itself can be monetized on its own. Pharmaceutical companies would love it because it's a huge data bank of hundreds of thousands of lines of information, reproduced many times with high reproducibility. And then in addition, to corroborate all of this, Rene's work has been scientifically peer-reviewed independently and funded competitively by no less than the NIH and the Department of Defense. These are highly competitive national fundings, which is an arduous process to go through. And he has achieved millions of rounds of non-dilutional funding, which we have used to get to market. So these 5 reasons give us the pillars on which we think you consider investing. I'll leave it at that. We'll go through the detail later. Rene?
Thank you, Peter, for the great introduction. So I just wanted to give you a little bit about my background. I have 3 entirely different backgrounds in chemical physics from the IIT. I have a background in genomics and also neuroscience. Mostly I spent all of my career in genomics and neuroscience. I've been a scientific reviewer for a large number of funding agencies, including NASA, that'll become relevant to the talk today. As Peter said, I'm funded by DoD and the NIH. And this year, we have been pitching all across the country, from California to Washington, D.C. This is a 5th one is coming up in the Longevity Investor Conference in Gstaad, Switzerland, which I'm very, very excited about, where we will be talking about our aging portfolio. So our company has a large number of disease portfolios. There's brain cancer, there's the substance use disorder, which is the opioid crisis, funded by NIH, chronic stress and PTSD, which is funded by DoD. The big, most exciting and most advanced portfolio right now is Alzheimer's and longevity/aging. And we also have developmental disorders like autism and schizophrenia. Today, I will use the Alzheimer's portfolio as an example of how we do our science, and then I'll get to progeria, and we will see the axis of longevity and aging emerge from that. So as business goes, there are 3 steps we take: scaling and validation of diagnostic markers, and ultimately the discovery of drugs that we take to the market. Everything in green is how we derive either licensing revenues or actually direct sales of drugs over a time period of 5 to 6 years. The ask today is for Alzheimer's and possibly aging preclinical work. We're asking for about 166 million in phase tranches of 10, 16, 27, 56, and 57 over 6 years. This will allow us to test 2 drugs all the way through clinical trials. And here's the use of funds across the years. It's preclinical work, phase 1, phase 2, and phase 3. And what is the actual problem we're trying to address? We're trying to address a problem that is so gigantic that there is no early diagnostic tests or even effective therapeutics for most of the brain diseases and disorders. What was the problem? The problem was the assumption that rodents would be a great model for the discovery of the causes and therapeutics for human brain disorders. That has, over 20 years, led to disastrous consequences, because right now we actually don't have anything for most of these diseases. And so we believe the rodents a decade ago were not going to be equal to humans. So in 2015, we predicted the need to have a really commercializable model. We actually built it. It's called Human Brain Models. We have a patent right now. And very nicely in 2023, the FDA has come to the same conclusion through the FDA Modernization Act, have said there will be no animal testing. They prefer to not have animal testing, and to address human diversity, both in gender as well as in ethnicity. So what do? We take skin cells from patients through a proprietary process. We make brain mimics. These mimics you can see on the right, they have all the canonical structures, the cortex, the retina, the spinal cord. They are close to greater than 98% identical to the actual human brain. That puts us in a very enviable position because we have now characterized this at a molecular level, the expression of genes for specific regions. And that's shown here in a semicircular way, where I pointed to the cortex, the other end is the spinal cord. And you can see all the genes that are expressed. We have done this at the protein level, where we have gone all the way around from the cortex to the spinal cord, showing that all of the key proteins that define those regions are actually expressed. So then we are now moved this to a commercializable strategy, which is to take skin cells from patients, derive stem cells, derive organoids, create a massive database of most of the major brain disorders, which is in itself a marketable product. And we can also do NMR profiling, and that allows us to look at early diagnostic markers, drug targets, and ultimately drugs.
Rene?
Yes.
Can I ask real quick, the process of taking skin cells of the brain disease and creating the organoid, is that a patented process?
The process of taking stem cells to make a highly differentiated tissue like the brain organoids is the patent, not for the conversion of skin cells to stem cells. That is available as a commercial resource from plenty of companies.
Got it. Okay, thank you.
Yeah. So now I'm going to take Alzheimer's as an example and walk you through the entire process. And here's what I'm showing you. There are 6 graphs here on the top, going from day 0 from the left to right, day 3, and all the way to week 12. And what I'm showing you is the expression of 13,000 genes that define the brain roughly, and showing you on each axis one organoid that is randomly picked. And what you see is every time we pick an organoid, the gene expression data is pretty much a straight line, which means it's highly robust and reliable, very reproducible. We've done the same thing for Alzheimer's. I'll give you 2 examples. On the top is a familial form of Alzheimer's, the bottom sporadic, and again, at different time points, and again, we see reliability, reproducibility, and robustness. These are the 3 important scientific foundations for doing good science. Then what we're able to do is take these organoids from normal people, and I'm showing you 4 genes as an example, and from those who might have Alzheimer's. And then what we look for in the yellow circles, in orange circles, are the differences in gene expression. They are considered the biomarkers of the disease. So what I'm going to show you now is that our model data is highly corroborated independently by patient data. The first one is our data is corroborated by 80 genetics. Here's a table of some 74 biomarkers identified in 2022 by genetics from thousands of patients. However, we filed a patent way back between 2016 and 19, almost 5 years before that, which showed that more than 50% of these genes are already identified by us. Astoundingly, we also said AD is a complex disease with ALS, FTD, and Parkinson's in our patents. That was the key finding in 2022. So there's a convergence of our science with that of others. Our data is corroborated by 80 postmortem studies too, and I'm going to show them in a different way. There are 4 models here going from left to right and top to bottom, 2 familial on the top, and 2 at the bottom, one sporadic, and one is the ApoE model. And what I'm going to show you is that in the brown circles are the genes found in postmortem brains. There are roughly 2,000 genes that are different between postmortem AD and those who are at the same age postmortem. And in the blue circles are the biomarkers we have, and in all cases, we have roughly about 500 genes that overlap. So that gives us confidence we are on track on looking at the right thing. We find the common biomarker amongst them. We're now able to derive diagnostic markers at birth. And we also are able to identify drug targets using sophisticated mathematical methods that I won't get into. So I'll stop here before I get into the novel AD therapeutics and take any questions that the audience might have or anybody else has.
So hang on. I think you're going to cover this, but I'm going to ask the question now and you can answer it later. These discoveries are impressive. Do they have applications for longevity and healthier aging? Also, illness such as cardiac problems, cancer, and dementia?
Very much so, Arthur. That's a very, very good question. And in the second half, I'll be talking about a model of progeria. The people with progeria, they die early at about age 16, 17. And the core morbidity that they have besides the brain is enhanced cardiac problems, as well as augmented proclivity for cancer. So our therapeutic strategy that we're building out will actually likely have therapeutic value that goes way beyond what we're talking about in the brain right now. So I'll get to that, and it's coming up.
Great, thank you.
Yeah, so the novel AD therapeutics is going to show you, and I'm going to show you a model in which I'm looking in the left side. You see 4 circles that are actually 4 genes that are expressed in a healthy brain versus ones that are expressed in a diseased brain as a way to express how things go wrong over a period of time during development. And so here is 4 genes that are going in a nicely synchronized pattern. That would be the genes expressed across development to make all sorts of different proteins in your brain. But this is what happens, we believe, in Alzheimer's, where they become asynchronized due to a variant in the gene or environmental or nutritional factors. And the DEG stands for differential expression of genes. And you can see in a given time frame, we're able to measure the differences that is shown in the red bar. And if you keep doing this over and over, as the healthy brain continues to develop and do what it does, you begin to see a fabric. It's kind of like a sweater that has been knitted, which is why I use the word space-time fabric over time. And it is as though you have pulled one of the threads in the sweater, and all of a sudden, the entire fabric is starting to fall apart. That is what we believe is going on in Alzheimer's, right from birth. So having said that, we are now going to show you that those DEGs in red, in our model system, we have 4 model systems: the sporadic, the APP, the PSEN2, and ApoE4. And you can see a string of 15 genes that we picked out of the 500 because they have clinical correlates that we can measure in the clinic, in a patient. And when we now add the drug, which is shown in the green columns, you begin to see that they are both in magnitude and in polarity. That is, if they go up, they go down. If they go down, they go up across all 4 models. They are the best described for ApoE, which has the greatest number of genes that have gone wrong, which is why it's one of the highest risk alleles that is there for Alzheimer's. Now, I'm just reducing those same 15 biomarkers and showing them in an expanded fashion to show you how we would use them in new clinical trials, but starting at a point called the MCI, mild cognitive impairment, where we are able to continuously track how these things are changing, and to address Arthur's question on the number 1 there is blood pressure, heart contractility, heart failure. So we are looking at things that can alter cardiac function too. And in the blue bar is how we typically do it now, memory and cognitive impairment being the primary way in which we assess the efficacy of a drug. So to explain this even better, we believe that at birth, you trigger a cascade of networks, and then at different points, you increase the nutritional deficiency and environmental insult susceptibility that is shown in these, what are called clades, but they are like the tributaries of a river. And then you start to see the early drivers of the disease, starting with accelerated aging, inflammation, immunity, hyperlipidemia, hypertension, and the late consequences, amyloid, tangles, and cognitive failure. So our whole goal is to go right up the river, as you'll see as a metaphor, and find the drug targets. And we can do this across any individual. We can stratify the disease across any person using their skin cells. And we are able to take and do very targeted clinical trials. I'm just showing you how we would spend the money across 6 years as we take this from preclinical efficacy, toxicology, and then phase 1, phase 2, phase 3. What is our competition, or what's the current status? The current status is the latest drug released. And here is, in their own words, what we are actually faced with Alzheimer's. Brain bleeds in 31% of the patients. That is frightening in itself. To make matters worse, there is now evidence that the difference between the treatment and placebo groups was only 3 points. In other words, the drug ain't doing much. So we are offering you a much better trajectory to address this thing more effectively by going upstream.
So, Rene, can I just ask one slide back? One more. I'm sorry, one more. So is the opportunity to identify the upstream anomaly one that has to be done by a test at birth or close to it to then intercept with these drug targets? I mean, from a practical standpoint?
From a practical standpoint, we want to make the test very simple. A mouth swab or a skin cell, or even a peripheral blood monocyte coming out of your blood from a blood draw. We are now capable of doing this from skin cells. We actually have seen that a subset of the biomarkers are dysregulated in the skin cells themselves. So in other words, it's going to become very simple and relatively easy to set in as a newborn screen or anywhere during your life to know if you have a susceptibility to Alzheimer's.
That's amazing.
It is, and it's unfortunate that this is how the disease is. It's a neurodevelopmental disorder. It had a misnomer of being age-related, and that's because it was a form of progeria, an accelerated aging from birth. Nobody calls progeria an age-related disease, yet it is aging. So is Alzheimer's. So we are correcting the biology and correcting the terminology used, and you just see it at a slower rate over a longer period to ultimately reach a similar endpoint. Does that make sense?
Yeah.
Yeah.
Would it also, I mean, it might be too early to answer this question, but if this test becomes available and someone's 65, is it too late?
It's never too late, because never too late to intercede with the drug. What you will miss out is the opportunity to actually correct the earlier points of companion intervention, which is nutritional deficiency and environmental insult. However, having said that, you could still eliminate the environmental insults, or you give your brain a chance to recover. You could still add and correct the nutritional deficiencies, and the plasticity of the brain is so enormous that I do believe you'll gain a lot by just doing those two things. You add the drug in there, and in combination, because the drug is acting ahead of nutritional deficiency and environmental insults, I think there's real hope that even at 65, you won't get a late onset Alzheimer's if we do this thing right and clinical trials work out. There are a few ifs here, but I think the strategy, I believe, is really built on a bedrock of solid science. And it just has to be scaled and validated across hundreds of samples. But we believe just from the data sets and the metrics we've shown you, this is very likely to occur.
Yeah. Peter, did you want to say something?
Yeah, very quickly. I'd just like to add, I think the important thing for us to understand is the three drivers of virtually most diseases, including cancer and Alzheimer's, are in these buckets on the slide on the left. The genes, which you're born with, which you really can't change. And then, if you like, the nature part of it, the environment to which you're exposed. And Rene can talk later if you ask him about which factors in the environment we think are involved. And then the nutrition that you have as a child and as an adult. Since we can't really alter the genes you're born with, we can, with a skin biopsy, test your proclivity, and then, as young as possible, intervene by changing the other two variables, nutritional and environmental. So we've got a three-pronged attack: the drug, the diet, as well as the environmental factors. Thank you.
Thanks, Peter. All right, so I'm going to move ahead. We were at clinical trials. This is the current status. It's not very promising. I'll leave it at that. And so this is our larger portfolio. I'm just reminding you, and I'm going to focus on aging, though our database contains all of the biomarkers, which is the soft bank we have. We have identified targets. We're moving ahead with drug development for each one of them. But I'll focus on aging and a disease called progeria, which has access in multiple things, as you'll see. So when we did our Alzheimer's model, one of the shocking findings was there were two genes, one's called Werner gene and the other one's DHX9. Both of them, when they are altered, cause progeria. But when we were seeing that their expression was altered in these model systems, telling us we were looking at a nexus of aging. So to confirm that is actually the case, we decided to ask, can we look at a disease called progeria and compare that to those from blue zone areas where they live healthily for a long time, where it's the epigenetic clock that determines your health. And so what we did is to make the progeria model, which is shown here in the column. And these are the canonical hallmarks of aging: DNA stability, telomere attrition, epigenetic alteration, and I won't go on the list. But the telomeres is a very key aspect of the whole thing. And you can see in progeria that it's altered. The red bars means it's different from those who are normal compared to those who have progeria. Shockingly, the Alzheimer's showed all of the same biomarkers were altered. So we went from 2 biomarkers to looking at, 14 times 2 to 3 biomarkers per category. So we became very interested in this and said, wow, if this is the case, can we do this experiment, which is called the Benjamin Button experiment? You've seen the movie, where he's born with progeria, but then becomes younger. So can we do a rejuvenation? So what I'm going to show you is this has become possible. In the third column is the Alzheimer's model, one of the models that is treated with the drug. And you can see 90% of those biomarkers both have reversed themselves towards a normalcy. So that opens the door to saying, yes, you can modulate aging. We've done it for progeria. We should be able to do it for anybody at any time. And that opens the whole story of, is aging natural? The answer is no. It is for some lucky ones, they live healthily forever. And for others who are born with the progerial genes, they succumb to it early. Peter, would you like to add anything?
No, I think that's a remarkable bit of science. If you think that three supposedly different diseases share the same genetic hallmarks, and that the drug can reverse all of the genes involved. I mean, if it was just one or two, it would be amazing, but this is the whole panel. And on the left side of this drawing are the complex, the 15 and 14 steps that involved in aging in all of us. So we're looking at something that in a way, if you regard aging as a normal phenomenon, you've got three sets of people, the rest of us who haven't got it, those with progeria, those with Alzheimer's disease and early cognitive decline, and then all of us can have these changes reversed genetically by the addition of a drug to the tissue sample.
Thanks, Peter. So to get on with it, so what I'm going to do is I'm going to use a metaphor that can communicate this better. I'm going to use the Mississippi. So for over 100 years, we've seen that the Mississippi floods, especially in the southern part of the country, there's the flooding from Arkansas in 1927, in Memphis in 2011, and our clinical strategies are much like sandbagging the rivers. We have really never understood what caused the floods in the first place. And so our strategy was to create a therapeutic dam across the river of Alzheimer's and aging. And so we believe that once you do that, once you put the dam, you bring everything back to normalcy. And that's pretty much what I just showed you in the earlier slide. But to remind you, the riverbeds that are flooding are downstream of where we want to build the actual dam. And when we do that, I'm going to demonstrate that. That's the drug, that's the dam, that's the cascade of events that are now starting to become normalized. And then all of a sudden, if you get to it early enough, you begin to reverse everything else towards normalcy, which is why we are showing this in green, and you're able to restore a normalcy. And that's the evidence we have with the drug that we can actually do that. Now, the drug has to go to clinical trials, must not have side effects and other things, but we can deal with that. At least we know the strategy is absolutely perfect. So I will now get back to some of the business side that we have made big strides in. We have an issued patent. We have a large number of patents covering a lot of the things I've already showed you and others that I haven't showed you. We've had non-dilutable financial runways from the NIH, from the Department of Defense. We have a number of other grants. We're going to submit up to $11 million to continue to do our work of de-risking. And what I wanted to say is, in summary, did you have anything to say, Peter?
No, I think you've got the picture now.
Okay, so as Peter mentioned, and that your genes alone don't determine your health destiny. It is genes, the environment, and nutrition. And we can mitigate at least two of them. The future of medicine would be not like in the past, which was reactive at the time of onset. The future, we would do all of this stuff in a brain disease in a dish and address them appropriately. And so the predictive strategy will be where we are all heading to. So now I wanted to touch upon something that I didn't touch upon last time, and I gave Arthur a little preview, and this is to do with the epigenetics of microgravity in aging and therapeutics. We know that our Senator Mark Kelly, he's senator now, he was an astronaut, and his twin brother, when he went up into space, his telomeres changed in length. And so it became apparent that microgravity was affecting the aging axis. This has become one of our biggest business models now, as we're being approached by both private and federal agencies to look and ask questions about, can you mitigate this risk? And we are very excited that Neurxstem will play a role in future space explorations and colonization of planets as we create our model systems to take into space long-term, assess risk, find drugs that can mitigate it. If you're lucky, the drug that we are now seeing with aging might also do it due to microgravity dependent aging. We don't know that till we do the experiment. And then that is going to be another pivot in our business model. And so I want to leave you with this incredible book that I've read a long time ago that very much fits with the way I think. We are all discoverers. Humans have been for thousands of years, and it's very elegantly a masterpiece written by Daniel Boorstin. But it was all about the discovery of Mother Earth. But we had just begun to explore space. Tomorrow, Chandran from the ISRO will land on the dark side of the moon. Why? Because there's water there, frozen. NASA has been exploring space for decades. And we are at an exciting point in which we're going to continue to explore and go into the future. And I'm just so proud and happy to say that we will hopefully help partner in the next phase of humans as the discoverers. And here are our distinguished people on our team. You met Peter, there's Clay Marsh at Virginia, there's Joseph Armstrong, who's originally from Merck, going to help us on the drug development side. And there's Dean Cole, who's a fractional CFO. These are our team. Susan was one of the co-inventors. Johnny and Manoj are business advisors. We have a phenomenal legal team, both Marshall Eisenberg, Melissa Hunter, and Kevin Noonan that help us with all our IPs. We are located in Heath, which is where SpaceX, Intel, Microsoft, and all have put big footprints down. We are the David in the big Goliath, so to speak, with the smaller group. And I wanted to leave you with this dark side of the moon. Some of us are old enough to remember Pink Floyd. And I just couldn't help leaving you with this phenomenal idea that they at one point were singing songs about it. Tomorrow, we're going to be actually exploring the dark side of the moon. And I'll stop there and take questions.
Super fascinating. Really, really fascinating. So relating to your comments about what happens in space to telomeres, right? Telomeres. It's a question. Oh, and I just want to throw in here, just because I'll name drop. I happen to know Roger Waters really well. So not only am I old enough, I've been able to spend time with him. He's fascinating guy. So here's a question. This sounds like significant financial potential meeting addressing growing longevity, commercial and healthcare markets around the world. And I think you've demonstrated that, but you want to comment a little bit more about that?
Yeah, so there's aging, aging in disease-related aging, which we can address, which is the dementia that we're starting to see this. We don't know where else we'll see this. We're going to try to change the trajectories of our biological clocks. Each of us has got a genome that will either make us live long enough or maybe much shorter than our full lifespan, our chronological lifespan. So the idea would be by addressing the three axes that help us live healthy and well. I'm not asking make you live, I'm not looking at immortality by any means right now. But what we're going to try to do is form your genomes from the functional genome. We'll be able to identify your risks from the environment, which will be different for different people, the nutritional issues you need to address. And at the same time, with and augmenting it with this drug, if you are, we might be able to move the needle forward for the vast majority of people towards a healthier aging, because it corrects for something that is, sorry, my phone's ringing. That'll correct whatever might be triggered by the gene variant that you were born with. And in space, that's a huge new exploration that we are undoubtedly going into low orbital space. This is the private industry is getting into it. NASA's getting into it, Defense Department. There's tremendous business opportunity there we would like to address. We're quite happy to partner with these incredible explorers that are putting their money to open a new future. So that's how I see it going, Arthur.
Got it. Peter?
Yes, thank you. Thanks, Rene, and thanks, Arthur. And if any of you watching would like to pop a question into the chat, Arthur will obviously ask it. We're here to have a conversation with you. Now, although we're both professors or former professors, we didn't want to lecture to you guys. I think one of the things to understand is that Rene's mind, is wired differently. I think he's a bit of an electrical engineer, electronic engineer. And instead of approaching things in a kind of single gene way, which is what most researchers do, one gene, one protein, one disease, he's looked at the whole sequence, if you like, the whole neural network and the regulatory network of large numbers of genes because of this technology that he has. So he's looking at hundreds and sometimes thousands of genes. And what he's able to see is patterns. And that's a novel way. Not only is the model novel and the patents novel and the SoftBank novel, but this way of looking at functional genomics. What are these genes doing? Which ones are lit up? Which ones are sleeping? How can you alter them at various life stages? And because of this novel way of thinking, this surprising discovery that let's call it normal aging, which all of us are going through, I mean, some of us more than others, is not a disease, but it is part of life. And it seems that the acceleration of the normal aging process, in the most extreme example, is what's causing progeria. And in other examples is causing that constellation of comorbidities, something that's polygenic, many, many genes involved. And then also the worst example is Alzheimer's disease itself. So you can see a continuum between born normal and then going through an accelerated process. If we could, for example, in a family where grandpa or grandma or father or mother had premature aging, we could look at the children, whatever age they are, in their 40s or 50, and we can look at the grandchildren at birth and see if they've inherited this cluster of genes which predicts later cognitive decline or Alzheimer's disease. And that gets us towards what we're really after, which is targeted, precision, personalized, preventive, all the P's, medicine. And this is the only example I've ever seen in all the hundreds of startup companies and so on I've been involved with, where this kind of much broader, almost Einsteinian, you can see this guy, Rene, is a physicist as well. And he has learned biology the hard way, worked for a Nobel Prize winner. And by thinking about the circuitry of what's going on, he's been able to nail the gene networks, and then using the simple Mississippi example of saying, well, the way to damn this thing is to go upstream and start preventing it from happening in the first place. And that's a very novel approach.
So super helpful. I know the clinical trials and all the work has to be done and side effects and efficacy and all that. Is it your sense presently, without backing you in the corner, that this drug would be applied initially and then over time to keep that dam in place? Or would it be something that could be implemented and fix the problem?
Rene, you might want to.
Yes, I will try to address this a different way. There are multiple modalities, therapeutic modalities by which we can try to address this. The drug, as you clearly point out, might need to be given periodically to maintain the networks towards a normalcy. However, we know what the target is. The target is not made public. And we might be able to correct using modern RNA therapeutics and other strategies the target itself. Once the target is normalized, I think you may not even have to do the drug thing. So I want to be very careful in saying we are not a one instrument band. We think broadly and we have many ways to do this. I just wanted to show you the evidence that using small molecules, we can accomplish this, but it doesn't rule out other therapeutic modalities that are coming into play right now, where we will be able to use a gene therapy strategy to address the underlying issue that gives rise to the load that is not functioning correctly. We can, if it's downregulated, we can upregulate it. When that corrects, the entire network will self-correct. We wouldn't have to do small molecules.
Got it. Peter?
I think what's very exciting about this work is the idea that by thinking about the function of the genes and the way the network functions. I mean, a simple example, which you'll all understand, there's an overlap between aging, obesity, diabetes, high blood pressure, cardiovascular disease, to name but four. And this is very much surprisingly analogous. In other words, the aging process is linked to this cascade of genes which are in a network. There are hundreds of them, which are linked to premature aging, to explain progeria, to early cognitive decline without Alzheimer's, to Alzheimer's in life, but also in death, when you look at the post-mortem brain. And what we've now analyzed is the whole spectrum from, if you like, from birth to death. And where Rene's been, also very smart, the drug is a repurposed drug. It's already been FDA approved for another purpose. It's safe. It's obviously going to be, we're going to want to use a trial for a different purpose, which we'll need FDA permission for. But it's highly likely to be both safe, because it's already been tested, and because Rene understands the network, he knows which drugs to pick. So, for example, if we were studying cholesterol, we would use a statin-type drug. If you were studying diabetes, you'd use a metformin-type drug. And so this is really targeting it in a logical way. Once you understand the gene network that's abnormal, you can see the much higher level of thinking that Rene is showing. He can more or less pull off the shelf drugs that we know will affect that particular part of the pathway. So this could accelerate the clinical trial process. We may not have to invent new drugs. We're almost reverse engineering it, saying, hey, we know the target. Let's look at which drugs affect that target. And there are plenty of drugs on the shelf, some of which may already be in use for other purposes. Some may have been discarded because they didn't meet the trial. I mean, you all know how Viagra was discovered, and so on. So I think it's going to be much more efficient and cheaper for humanity to get the answers in a practical way within our lifetimes by repurposing drugs through careful targeting, by understanding the genes that are involved and the networks that are vulnerable.
I just want to, sorry, Arthur, I just want to say one thing. The drug that I showed you is not one that is FDA approved. However, there is a drug in the same category that is FDA approved. So we will test both of those. If the one that is FDA approved, exact same class, everything else, same target, if it does what it does, we will jumpstart the whole project, because we go right past the safety and everything else and can move it into clinical trials.
Yeah, so it compresses the time, right?
Compresses the
That's
time.
right.
Yeah.
Yeah.
The second thing I want to tell you, I'm so much more excited about something that I didn't talk about, which is cancer. We now have a database of cancer and the network of cancer. Unbelievable. And we are processing that information by interrogating the system to understand how it behaves. That's number one. The second thing is, from the progerial model, we know that they have increased rates of cancer. So by combining these two models in the data sets, and at the interface of those two things will emerge entirely new ways to think about the most daunting of all diseases we all face is cancer. So I, as a scientist and a theorist and everything else, love that challenge of breaking into places where we have had very little success. And our strategy, hopefully in the future, would be to make it into a completely manageable disease at the very least, or a completely cured disease at the other extreme. So when you join our company and back us, you're going to be getting into so many more things than just what we are now saying is the best business model. I just want to leave that as a wide open possibility. The space is another one. So we're very excited to be sharing this publicly. Got the opportunity to invite people who want to change the future. We have the means to do it. We have the science, back us, let us take a shot at it.
Yeah. Fascinating stuff, and really appreciate you sharing all this new, really vital information. And there's lots of links and information available that we'll continue to share with you of the opportunities where Rene has been able to speak with the science community and invited to do so because of how remarkable this approach and technology is. So, not only do you have some of that stuff now, we'll make sure that you have it going forward and put you in direct touch with Rene and company here post webinar.
Yes, so you do know, and I think it's on there. We have two very exciting opportunities. One is in Gstaad, Switzerland, end of September, where I'm talking to a very curated audience on longevity. And then I'm in the Lausanne X initiative, which is an international initiative using both policymakers as well as investors. That is the first week of October in Lausanne. And I'm a special invitee over there. So there'll be a new opportunity to share this across a much wider audience in Europe as we all collectively make our efforts to change the future for all humanity. So I think that's what you were trying to allude to. Yeah,
exactly.
And so it's another bunch of exciting meetings coming up, meeting new people, explaining the science all over again. So thank you, Arthur. This has been a phenomenal forum for us. I've enjoyed every one of our meetings, and
as I have.
Wonderful host.
Yeah, I super appreciate you both being here. And there's a bunch of questions we didn't get to, but we'll make sure that we diary them for you to answer independently post-webinar. And super appreciate it. Fascinating stuff, really, beyond changing the world. So really glad you're here again. And I'm sure we'll do this again soon. With that, as I said, if there's anybody in your world that would be interested in learning more, we're going to make sure we can cause that to happen. And then we have the recording. And as I always say, thank you for sharing with us. The only thing you can't make more of, and that's your time. So till next time, thanks, guys.
Thank you all for the audience.
Thank you all. Thanks very much.
And
thanks,
Rene, I'll follow up with you. Okay.
Yes, thank you.
Thank you.
Thanks, Peter.
Bye now. Bye.